Thank you very much. So my name is Felipe. I'm a PhD student at the BO Lab in the Center for Genomic Regulation in Barcelona, Spain. And I'm going to share with you the first results of my PhD project that aim, among other things, to assess how different microbiome profiling approaches can detect host genetic effects in the gut microbiome. So why do we target the detection of host genetic effects in the gut microbiome? So we all know that there are many associations that were shown in the past between the gut microbiome and the host complex diseases. And we also know that each one of us have different gut microbiome compositions, which lead us to think, okay, can these different microbiome compositions can be caused by our unique genetic makeup? So if we put all these things together, we can believe that if we understand how the host genotypes is influencing the gut microbiome composition, maybe we can start to understand how the microbiome and the host is interaction and how we can connect this with the complex diseases to see whether the gut microbiome is mediating some host genetic effects into these complex diseases. So this is very important for us. However, very few strong genetic effects were detected so far in the gut microbiome. And when we talk about the gut microbiome composition, I'm talking about the relative abundances of different bacteria that live in this community. And because of these very few strong associations, we started to wonder whether this is actually a biological fact or if this is happening because limitations of the current profiling approaches. So when we talk about approaches, the first thing is to think about the different sequencing method. So what people normally use to profile the microbiome is the 16S single marker gene, which is very good for us in terms of genetic epidemiology, because if we want to work with a lot of individuals that are genetically diverse, it allows you to work with this large population because it's not very expensive. However, it has many limitations based on PCR biases. Few bacteria have multiple copies of this gene. And the main problem is that we cannot have a very good resolution on the level of the bacterial species, only on the genus level. And another thing is that because we are only mapping one gene, we can only infer what are the other bacterial genes present in a microbiome based on the taxonomic profile that we did before. So we can only infer, we cannot directly map these genes. And we also think in terms of different approaches that mapping bacterial functions instead of taxa can also reveal more and stronger host genetic effects, because we know that bacteria exchange DNA a lot, and also there is a lot of functional redundancy between different species. So we also think that having a method that can allow us to map bacterial functions directly could be a good option. So on the other side, we have the deep shotgun sequencing, which is super good in terms of mapping genes directly. We can go to not only on the species level, but on the strains level. However, it is so expensive that it will be difficult for us to work with a large population of individuals. So in our project, we are proposing using shallow shotgun sequencing as an alternative because it is not as much as expensive as 16S. It is, very cheap compared to deep shotgun data. However, even though we can review direct mapping of genes and it can go to the species level, just like deep shotgun, we can only work with this method if we have a very good reference catalog of microbial genomes, because we cannot reassemble the genes and the genomes of the bacteria that are mapped by our metagenomic data. So using a reference catalog is very important for us. In my project, I'm working with shallow shotgun data and 16S data from NIDA's HS rats. I'm using data from the cecum of those rats. And all these rats were also genotyped for more than 3 million SNPs and phenotyped mainly for drug addiction and metabolic diseases. And these animals are coming from two different phenotyping centers, one from Michigan and one from New York. And for the shallow shotgun sequencing, we targeted 1 million pairs of short reads per sample. I'm not going through the details of the pipelines I used, but just for you to understand. So I used different pipelines to get the taxonomic profiles using 16S and shallow shotgun data. However, for both of them, I was able to use the same taxonomy annotation, which was important for further comparisons. And as you can see here, for using the shallow shotgun data, I was able to recover much more species. I was able to map much more species than 16S, which was expected by us based on what I said before. And one thing that is important to highlight here is that because there is no rat-based microbiome genome catalog available for us, I had to use a very broad microbial catalog, which is GTDB, that has bacteria coming from different hosts or even from different environments. So this is a very big catalog, and there were many adaptations we needed to do, both to use shallow and to use this broad catalog. Here, just to give you a glimpse of how I did the functional profiling, I used a catalog based on protein families, and then I collapsed the counts of reads for each protein family to eggnog functions. And in the end, I used every one of these profiles, which are basically tables containing the relative abundances of each one of the taxa or functions by sample. And I used the host genotypes that we had to run a host genetic effect analysis, a heritability tests, and GWAS using a linear mixed model. So the results I have for the aggregate heritability test showed me that shallow shotgun data was not even not only capable of mapping more species, mapping more taxa, but also it was able to review more taxa that had significant heritability, as you can see here on the table on the right side. But if I only get the taxa that are common that were mapped for both methods, you know, the taxa that are common for 16S and shallow, we can also see that the heritability, most of the heritability values for this common taxa tended to be higher when I had these results based on the shallow shotgun data compared to 16S. So this is a very good result showing that maybe shallow shotgun data is a good alternative compared to 16S. In terms of GWAS, what we had was after all this Bonferroni adjustments for correction for multiple sampling, we were able to find the same number of significant taxa. However, it is important, it is super interesting to see that for 16S, as I said, we can have a good resolution on the genus level. The strongest association was with the genus Paraprevotella, while for shallow shotgun data, the strongest association was seen for one of the Paraprevotella species, which is going to be important for us because if we know the species, we can map, we can track the genome of this species and try to find what are the bacterial genes there that can be, responsible for this association. One thing that was, not very good was the result for the functional profile. So I was not able to recover any significant biological functions in terms of heritability. And what we think is that for functional profiling, because we are mapping individual bacterial genes, maybe shallow shotgun data is not very appropriate for the direct mapping because it tends to be, the functional profiles tend to be more sparse than the taxonomic profiles. So in the end, I end mapping the most abundant genes better, which are the genes that are more associated with central biological functions, which are, the same for all bacteria. So one solution for that is to also do prediction of functions based on taxonomic profile. But as we see that shallow shotgun data can have a higher accuracy, better results compared to 16S, we also think that these predictions can be better using shallow shotgun data instead of 16S. And for that, working with a very with a host-specific catalog could be super important for the success of these predictions. And I'm going to show you how we're going to plan, how we're planning to do it. So the next step of my project is building the RetMag catalog. And I'm already doing that in collaboration with NIDA and Magnify. We're working with 96 rats from the HS population, and we are working with many different samples. We collected cecal and fecal samples. We worked with rats coming from 7 different phenotyping centers, different ages. Also, those rats, they were submitted to different conditions, so they were fasted the night before the collection or not. They are naive or they were phenotyped for drug addiction. All of this to maximize the microbiome diversity. And as you can see here on the right side, we performed microbiome diversity analysis in order to see whether these different factors were could explain differences in terms of diversity, and we saw that these factors were really helping us to get a higher diversity that we expect to reflect in a high number of different diverse genomes that we can recover from the metagenomic data we are collecting. So for this, we are sequencing very deeply. We're targeting 50 million pairs of reads per sample. And the idea is to use this metagenomic data to reassemble the bacterial genomes and to use this genome catalog as a reference to run the same pipeline and see whether we can improve the taxonomic profiles and also use these for functional predictions. So in conclusion, as you can see, the shallow shotgun data seems to be a very promising alternative to 16S in order to detect host genetic effects in the gut microbiome. We saw that very low single gene coverage might be behind the poor results of shallow shotgun based functional profiling. And we also believe that the adoption of a rat gut microbiome catalog can not only improve the results for taxonomic profiling, but also be used for the inference of what bacterial functions are there. So we can run functional profiles and see whether the host genetic effects are indeed stronger on the functions rather than taxonomy. I would like to thank Amelie Boat, my supervisor, the rest of the Boat Lab, also our sponsors and our collaborators. Thank you very much.